All right, welcome to the Nova Institute project. So glad you're here with us. In this explainer, we are diving deep into the absolute chemistry of life itself. We're going to look at the fundamental building blocks that make up every single living organism on this planet. We're getting into not just what these molecules are, but how their highly specific shapes dictate exactly what they do.
So let's kick things off with a pretty massive question. What exactly makes a living thing, well, living? I want you to think like a biological detective for a second. If we zoom all the way down, past the organs, past the tissues, and even past the cells, we find this wild, microscopic landscape of chemical ingredients. But how do those lifeless chemical elements actually combine to create a breathing, surviving organism? What are the foundational chemical clues we should be looking for?
Part 1. Carbon, life's ultimate backbone.
Think of carbon as the ultimate biological Lego baseplate. It's the central hub everything else snaps onto. It has this literally perfect setup to form exactly 4 stable bonds. It's constantly shaking hands with hydrogen, oxygen, nitrogen, and phosphorus. Those specific chemical handshakes are what allow carbon to build the massive, complex molecular structures that are absolutely crucial for life. Honestly, without this 4-bond capability, the whole shebang, all those complex macromolecules we study, they just wouldn't exist.
Part 2. Meet the 4 macromolecules.
So, if we're investigating living things, everything really boils down to 4 main suspects. We've got carbohydrates, lipids, proteins, and nucleic acids. Every single organism on Earth, from the tiniest bacteria in a petri dish to a massive blue whale, relies on this exact same core lineup. Let's break them down one by one and see how their unique shapes give them such specialized jobs. First up, carbohydrates. These are built purely from carbon, hydrogen, and oxygen. Now, the big takeaway here is how they snap into these distinct ring or chain formations. That specific structure is incredibly efficient for storing quick grab-and-go energy, like glucose. But hey, they aren't just a quick sugar rush. When they link into long, complex chains, like cellulose, they are incredibly tough. They literally build the rigid cell walls in plants. Form directly equals function. Next, we've got lipids. Now, these are mostly nonpolar hydrocarbons, which is just a fancy science way of saying they seriously hate water. That hydrophobic nature is absolutely crucial because it makes them perfect for building cell membranes. They act like molecular bouncers, securely controlling exactly what gets in and out of a cell. On top of that, lipids are the ultimate long-term energy vault. Triglycerides actually pack way more energy than carbs do, and they provide vital insulation to keep organisms warm. Moving on to the heavy lifters, proteins. These are the ultimate biological multitaskers. They start out as simple chains of amino acids, but the real magic happens when they fold. They fold into these wildly complex, specific 3D shapes. And that precise shape, it dictates exactly what they can do. They act as enzymes, speeding up chemical reactions. They act as transporters, lugging oxygen around in your blood. And they provide structural support like the keratin in your hair. Honestly, if the shape of a protein breaks, its function completely breaks. And finally, nucleic acids, the master instruction manuals of life. These are built from individual units called nucleotides, which is just a sugar, a phosphate, and a nitrogenous base. You know the famous double helix structure of DNA, right? Well, DNA hoards all the genetic instructions needed to build an organism, while its close partner, RNA, actually transmits that data so the cell can get to work. They are literally the data storage and transmission centers of the entire cell. Part 3. How structure drives function. So tying it all together, the structure of these molecules isn't just random. It completely defines what they can pull off. Rigid carb rings mean energy and structural support. Water-repelling lipid chains mean secure membranes and long-term storage. Crazy folded protein ribbons mean custom tools and transporters. And tightly coiled nucleic acids mean genetic regulation. Finding any of these macromolecules in a lab sample is basically a smoking gun for the specific biological functions that organism is capable of. Part 4. Evidence versus biological inference. Okay, let's talk logic, because it is so easy to make a bad leap here. Detecting a molecule gives us hard evidence, right? Not the entire biography of the organism. Good sound logic is saying, hey, we found protein in this sample, so this organism uses proteins for cellular functions, like making enzymes. That's solid. Bad logic, and we see this happen all the time, is finding a protein and immediately jumping to, wow, this organism must eat meat. No way. Plants have proteins. Bacteria have proteins. You absolutely cannot assume behavior or diet from a microscopic chemical footprint. So I need you to burn this rule into your brain. Detection equals presence. It does not equal behavior, and it does not equal diet.
Whenever you find a macromolecule, the only question you should be asking yourself is, what cellular function does this molecule serve? The evidence has to drive the bus, never our assumptions.
Part 5. Solving the lab case. All right, let's put this into practice. We've got a fresh, unknown biological sample straight from the lab, and we need to figure out its biological capabilities. Let's use our new framework to decode what this organism can do, based purely on the chemical fingerprints we can extract from it. Here's the logical breakdown. Step 1. We run the tests and boom, we detect lipids and proteins. Moving to step 2. What does that actually imply biologically? Well, lipids mean we've got cell membranes present. Proteins mean we've got active enzymes ready to do work. Finally, step 3, we deduce the actual life functions. Thanks to those lipids, this organism can handle membrane transport and store energy. Thanks to the proteins, it's doing enzymatic reactions and cellular regulation. We just went from basic chemical swabs straight to biological capabilities, just by strictly following the evidence.
But hold on, it is time for a massive reality check. Just finding these macromolecules does not guarantee that the organism is surviving or even currently alive. Seriously, a piece of dried up firewood is packed with carbohydrates, but it is definitely not a living, growing tree anymore, right? These molecules are the required ingredients, sure, but their mere presence doesn't tell us if the engine is actually turned on and running. Which brings us to a pretty wild, provocative thought to wrap up today's explainer. If carbon-based macromolecules, the carbs, the lipids, the proteins, and the nucleic acids, are really just the physical, inactive ingredients in a biological recipe, what is the spark? What else is fundamentally required to turn that chemical soup into a living, breathing, functioning organism? Think on that one as you review your notes. Thanks so much for joining me today. Stay curious, trust the evidence, and I'll catch you in the next lab.